Hello, my name is Marvin Petersen. I'm a medical doctor as well as PhD student at the University Medical Center Hamburg-Eppendorf. And on behalf of my colleagues, I'm very happy to talk about the topic a latent clinical anatomical dimension relating metabolic syndrome to brain morphology and cognition. Metabolic syndrome is a composition of multiple cardiometabolic risk factors, including obesity, arterial hypertension, dyslipidemia, and insulin resistance. With a prevalence of 23 to 35%, reported in Western societies, it is a very common condition. As it increases the risk of stroke, cognitive decline, and dementia, it poses a relevant neurological public health challenge. However, its neurobiological underpinnings are not very well understood. Previous analyses have shown that cardiometabolic risk is linked to brain morphological abnormalities. However, this association still is lacking in terms of understanding because certain characteristics of these analyses. Typically, they lack resolution and scope, being focused only on global morphometric measures as well as singular risk factors. They are typically in a group design, disregarding potential continuous effects, and overall have low sample sizes, which has implications for their reproducibility. We addressed these points, performing a surface-based cortical thickness analysis. Our analysis was based on a pooled sample from the Hamburg City Health Study and the UK Biobank, totaling a final analysis sample of 40,087 individuals. We investigated from these two samples the cognitive data, the metabolic syndrome component information, for example, the blood pressure measures, as well as structural MRIs. On the structural MRIs, we performed surface-based morphometry harnessing CAT12 to derive cortical thickness measures on a regional level for Schäfer 400 parcels. We associated the cortical thickness measures with the clinical information by employing a partially squares correlation analysis. Now, a partially squares correlation analysis, or PLS, is a multivariate data-driven statistical approach, which is pretty similar to canonical correlation analysis, which you might be more familiar with. It takes into account two data domains and tries to identify latent association in this data. In our case, the morphometry data being a multivariable dataset subjects by Schäfer 400 regions, as well as the clinical data being a subjects by clinical scores dataset or matrix. Now, the PLS tries to identify now the latent association within these datasets by deriving an imaging covariance profile, or imaging loadings and clinical covariance profile, which describe the overall association between the data. Furthermore, we get subject level scores, an imaging score, as well as a clinical score, which show for each individual covariance profile and each individual subject the adherence to these covariance profiles. Together, the imaging and clinical covariance profile and subject scores are called a latent variable, and PLS tries to identify multiple latent variables which associate the input data. We assessed the significance of the modeling approach as well as its robustness via a permutation and bootstrapping analysis. Furthermore, the inputs were residualized for age, gender, education, and cohort to address these effects as potential confounders, to make sure only metabolic syndrome effects are really identified in this analysis. So here we see the PLS results. Here in this scree plot on the upper left are shown all the latent variables investigated. The PLS successfully identified 7 significant latent variables, which are shown on the x-axis, here shown by the gray plots below the scattered line. The first latent variable explained the largest share of the variance, 77%, and was subjected to further analysis. It was associated with a clinical covariance pattern and imaging covariance pattern. Overall, the latent association or latent variable represented an association of a widespread cortical thickness abnormality pattern, being highlighted here in colors, and metabolic syndrome severity. In blue regions, higher cortical thickness was associated with lower MET severity. In red, higher cortical thickness was associated with higher metabolic syndrome severity. If we take a closer look at the clinical covariance profile, we can see that the covariates do not significantly contribute to its association, as their confidence, as their overall loadings are low, close to zero, and the confidence intervals are cutting the zero line. Confidence intervals were derived from the bootstrapping analysis. Most strongly contributing were the obesity measuring variables, for example, waist circumference and hip circumference, and which tells us potentially that that obesity might be the most parsimonious explanation to the association found. Furthermore, the blood pressure measures, dyslipidemia measures, and the insulin resistance measures were also significantly contributing. So we're also interested in the role of this association in terms of cognitive function. So we reperformed the analysis within the subsamples, here shown for the UK Biobank. We simply added the respective cognitive variables, which were differing between both cohort, the UK Biobank and the HCHS. And by doing this here, for example, for the UKB, we could see that to this latent association, to this latent variable, were also the cognitive variables significantly contributing. So taken together, we identified a latent association relating metabolic syndrome to cortical thickness abnormalities, as well as cognitive performance. Now, we are also interested in the determinants of this cortical thickness abnormality pattern I've shown you. For this, we performed a virtual histology analysis. We took regional gene expression profiles from the Abogen toolbox, which stem from the Allen Human Brain Atlas, and derived maps of cell-specific transcriptomic signatures, so cell density maps for astroglia, oligodendroglia, microglia, excitatory and inhibitory neurons, as well as endothelial cells. And these maps of cell densities for different cell populations were related to the imaging covariance profile on a region-by-region basis, performing a spatial correlation analysis, addressing via statistical approaches the problem of spatial autocorrelation. This analysis was performed based on the AppAnnotate toolbox from Leon Lotter and Jürgen Dukat. Here are the results of the virtual histology analysis. What we found was that the imaging covariance pattern, as shown here, was significantly associated with the density of endothelial cells, microglia, as well as excitatory neurons. Here you can see further neuron populations being significantly associated, but these were not reproducible in the sensitivity analyses. So we could see that metabolic syndrome effects were strongest in regions with high endothelium, high microglia amount, as well as high density of excitatory neurons of subtype 8. Now, we deem this very plausible, as histopathological studies have shown that endothelial dysfunction as well as microglia activity are implied in the overall pathophysiology of metabolic syndrome effects on the brain. And furthermore, excitatory neurons of subtype 8 entertain long-ranging corticothalamic projections, which makes them potentially susceptible to small vessel pathology, which is prompted by metabolic syndrome. So we think this finding is plausibly explainable as well. Now, to summarize, we performed in 2 population-based cohorts totaling a sample of 40,087 individuals, a multimodal neuroimaging analysis based on data-driven and multivariate statistics. These approaches helped us to identify a latent clinical anatomical dimension relating metabolic syndrome, cognitive function, as well as brain structure. This dimension explained most of the covariance of this association. The overall association was found to contribute to a widespread pattern of cortical thickness abnormalities, which was determined by the cellular density of endothelial cells, microglia, as well as neurons with long-range connectivity. In further SIP-analysis, we could show that it seemed to also be associated with brain network topology. If you're interested in this, as well as further details on the analysis, I'm very happy to encourage you to take a look at the preprint, which is on bioRxiv. So just follow the QR code. I want to thank all the colleagues involved in the project from our working group, as well as a working group of Kaustup Patil, Simon Eickhoff, and Felix Hofstetter from the Forschungszentrum Jülich. And with this, I want to thank you for your interest.